Hello everyone, this is Rafi and welcome to my channel. Today we are going to discuss a very interesting paper, which is identification of novel diagnostic and prognostic gene signature biomarkers for breast cancer using artificial intelligence and machine learning. I don't know why they use and machine learning because I think machine learning, artificial intelligence is a umbrella term in which we use machine learning as a method. So assist transcriptomic analysis. Transcriptomic analysis means they might be using gene expression dataset, and it's look like they are using different machine learning algorithms to find out the most relevant biomarkers by comparing gene expression between patient with breast cancer with control samples, and more likely the samples will be a tissue samples which is extracted after performing the biopsy, and the control sample will be the same sample. I mean, from the same patient, but from the region where there will be no cancer cells. So let's see. Let's go to the methodology. What they have done. So they said the raw gene expression data set. So they use gene expression data sets, and this is the raw format of gene expression data set, which is cell format. And they have used 11 data sets, and they have mentioned the IDs of these data set, and they are using the NCBI Geo website, which contain the gene expression data sets from microarray, RNA-seq, and ChIP-seq, and other lot of technologies. So they have used Geo database, and they are using jQuery package, which is external package in you can download into your R programming language, R programming software. Using the jQuery, they extracted the uh, data of these IDs. So so they have some inclusion criteria in which they use human breast tumor samples. The sample will be from tumor tissue, and they have used around 701 samples, in which 356 are breast cancer tissue samples and 345 are controls. So in the first step, they have performed some pre-processing because when we have cell files, we need to do some normalizations. So they use the package Oligo, which is also an external package in our software. After that, they perform all the traditional methodology like creating PCA plot, principal component analysis, using our software. And these are the data sets, and what normalization method they have used is mentioned here. After that, they have used Lima package, which is also available in R as an external package. So this software or this package is used for performing the differential expression analysis. Actually, what this software do, this software compare the gene expression of cancer group with control groups, and then we have figure out if we get, because after performing the gene expression data analysis, we get the results in log FC. Log FC means fold change, how much gene expression increase or decrease. If the gene expression or log FC is plus or plus 2 or more than that, and p-value is also a significant, which is less than is equal to 0.05, then we can say that this expression is highly significant. It's mean the gene expression might be high in tumor samples when we compare it with the control samples. And if we get log FC value in negative, like more than -2, or p-value is also significant, which is 0.05, then we can say this is also a significant result. But in this scenario, the gene expression uh, is high in control group as compared to the tumor sample. So this is also an insignificant result. This particular gene is expressing high in control group, but after getting the breast cancer or any other tumor sample, after getting any tumor, the tumor uh, uh, decrease the gene expression of a particular gene in tumor samples. So this will be also a good result. Uh, After that, here is the cutoff they have used. After gene performing gene expression analysis, they have used the cutoff of, for example, p-value must be less than 0.05, and the log FC or log 2 fold chain must be greater than +2 or -2. So after that, they have also performed functional pathway analysis and gene set enrichment analysis, in which we have to figure out in which pathway is gene is involved, because we will get the list of significant genes, and we want to see in which pathway these genes are involved. And after that, they might get some sort of list in which they have the significant number of genes which they have found significant in Lima software. After that, they have applied machine learning and feature selection method. In machine learning, we have to select some features. Features are like, uh, for example, in this scenario, genes are the features. Uh, we are predicting the genes, which is highly significant. So in this scenario, genes are the features. And in machine learning, they are using, I think they are using different type of, yeah, here they have mentioned different, around 5 different machine learning algorithms they are using. And after that, they have used couple more, around 5 more machine learning algorithms uh, to validate the results. Okay, so these are some descriptions of the machine learning algorithms they are using. Actually, machine learning algorithms are different type of approaches, or in some easy explanation, you can say there are in machine learning there are different type of methods or different type of formulas to calculate the relevant results. So there are so many type of algorithm available in machine learning. So they are using around, I think, 10 different machine learning algorithms. So this might be they are using 10 different algorithms because after getting the results of these 10 different algorithms, they can say these genes are found in 6 different algorithms, and just this gene is found in 8 different algorithms. So this will also increase the significance of the gene because you are getting the same gene in different 8 different algorithms or more than 8 different algorithms. I mean, there you are getting significance of that gene in multiple algorithms. So you can say this gene might be have any important role in breast cancer. Uh, so After that, they have also performed survival analysis because they might have the data of patients. In most of the time in survival analysis, we need the patient last follow-up date. So we will see what is the last follow-up days date. So we'll found out the total number of days after the after performing the biopsy, and we will see what will be the outcome if the patient is alive or dead. So they have used the survival analysis to get the uh, relevance or get the uh, importance of the time in which you will get what will the end result of the uh, breast cancer. After getting the significant biomarkers, they have they have actually validate their results in wet lab. So they have collected the breast cancer tissues from the patients, and then they have performed RNA isolation and performed QRT-PCR, or you can say real-time PCR. So these are the plots which shows the normalization of gene expression data sets, and these are the genes. In left side, you will get the gene which have lower log FC value, mean these gene increased in control group when compared to the tumor group, and these genes having fold chain +2 means these genes is highly expressed in tumor group as compared to the control groups, and these genes are non-significant. And these are the list of top 10 upregulated or downregulated differentially expressed genes. They might get it after applying the bioinformatics tool, which is Lima in R. And this is the pathway analysis of these genes. After performing the machine learning, they have calculated they have performed cross validation in which you will figure out what is the performance of your machine learning algorithm. So here they calculate the AUC value or accuracy, precision, recall, or F1 value of each algorithm. These are the list of 5 algorithms. So all the algorithms looks good because they have around 90 more than 95% AUC or accuracy, precision, recall, or F1 score. So the algorithms work very well. So here is the list of 28 genes they have found highly significant or non-significant in machine learning algorithms. And here is the KM plot, Kaplan-Meier plot, which is the survival curve plot. And here is the gene which they think is highly significant, and which gene is upregulated and which gene found downregulated after performing the survival analysis. Here are some more cross-validation ROC curves, and here is the result of experimental analysis which they have performed in lab using qPCR, and here's the result of qPCR. So let's find out what they have found in conclusion. So they said initially 355 differentially expressed genes is selected in bioarray data, in which these genes are go through the machine learning analysis. As a result, two novel gene signatures, nine gene signatures is used for diagnostic, and eight gene signature will be used for prognostic. They have found these two sets using machine learning algorithms, and they have also performed the qPCR of these two set of genes. So the approach is very good because in this approach they have used the bioinformatics in the form of differentially expressed gene analysis using Llama, and after filter out the genes from the Llama package, they have applied different 10 different type of machine learning algorithms, and after that, after getting the list of those genes, they have performed uh, experimentation validation of those biomarkers. So thank you so much.